and today I'm going to give you a talk about structure-based drug discovery in biomedicine. So I thought I would just start with a little bit about me. So this is a picture. This is a very old picture of me actually, because I used to have very short hair, but this was when I first started at Oxford. Um, but basically, I graduated back in 2022 from the University of Southampton with an integrated master's degree in biochemistry. And now I'm studying at Oxford in collaboration with Diamond Light Source, doing my PhD in interdisciplinary bioscience, but it's a little bit more focused on drug discovery and also time resolved crystallography of some proteins that are involved in COVID. And so I guess that's why I'm qualified to give the talk. Um, and so I thought I would start with giving just like a basic overview of what we mean by structure-based drug discovery, because maybe we don't really know what we mean by structure. So in biology, but there's a field called structural biology, and this is basically the study of the 3D structure of biological molecules. So this could be anything from a protein to a nucleic acid like DNA, RNA, or some sort of carbohydrate. And from understanding the structure of these, like something like this, we can infer often the functions of the protein. And using these structural insights, we can get an idea of ways we could modify the function, or ways that we could affect the possibility of the biological molecule from doing its sort of activity. And so that's kind of the idea behind structure-based drug discovery. We want to use these structural insights that we have to get an understanding of how we can modify perhaps proteins that are involved in disease, and possibly find new drugs. And so there are a few different ways that we can get this sort of information. There are three main biophysical techniques, I would say. And these are X-ray crystallography, which is my favorite, um, NMR or nuclear magnetic resonance, and then cryo-EM. I'm sure you guys have probably learned a little bit about these, but I'll give a bit of an overview of each of them in turn. Sorry about that. Let me turn that off. Um, and so the main thing that sort of sets these different structural biology techniques apart when deciding on how you're going to study your biological molecule is size limits. And so with NMR, you can only really look at small proteins or small biological molecules. Anything below 40 or 50 kilodaltons is usually good for seeing in NMR. Whereas with cryo-EM, you usually want bigger proteins. So anything that's above 36 kilodaltons is usually what they say is sort of the size limit for that. Whereas for X-ray crystallography, you're not particularly limited by size, but you're mostly limited by crystallization of your molecule. It's quite difficult to crystallize things. Trying to get them to form ordered structures, you need to find particular conditions, and often it can be quite difficult to narrow those down to something that's really gonna be perfect for that. And even if you get crystals, maybe they won't diffract on the beam. It's a whole thing. And yeah, so usually there's no size limit, but it's just about trying to find something that will actually crystallize. And so a little bit more about each of these techniques. With NMR, this is a technique that uses radio frequencies to excite atomic nuclei. And then once they're excited, they will relax, and the radiation that they give off when they relax back to their normal energy state can be used to understand the atom that's involved as well as their environment. And so this kind of gives us an idea of what's actually in the molecule, as well as what is surrounding each of these atoms. The processing, I would say, is probably the hardest part of NMR, because, you know, if you're looking at a large biological molecule, something like this is a big beta-barreled protein, you will end up with huge amounts of peaks on your spectra. And trying to deconvolute this and assign them to particular residues within the protein can be very, very difficult. Back in my undergrad, I did a short project on assigning the spectra of, I think it was a 8 residue protein, and it was very difficult trying to find ways to associate residues with each other and understand their chemical environment by hand is, yeah, it's a challenge. But luckily there are some programs that can do this now. And especially with the use of AlphaFold, you can kind of have an idea of what your spectrum might look like, and then sort of use both of these models together to get a true structure from your NMR. But yeah, like I said, it is limited by size, and so usually people don't do NMR if they're looking for high resolution structural data. In recent years, actually, cryo-EM has mostly been the technique that people look into. I mean, they won the Nobel Prize for this freeze quenching step or flash freezing step in cryo-EM. I think it was back in 2016. Maybe it was a little bit later than that. But cryo-EM basically acts to bridge the size limits that we have between NMR and crystallography. And so we kind of get a big range of biological molecules that we can look at now. But I'll just kind of take you through how we would set up a cryo-EM data collection, and then give you some real data from what I've collected from my PhD. And so the first step in cryo-EM is making your sample. Because you're looking at your sample in solution, you can look at a big range of things. You could look at full viruses, you could look at full cells, you could look at large complexes of proteins. Alternatively, you can just look at the protein by itself, and then you'll have it in a nice solution, hopefully something that doesn't have a lot of other molecules in it, and something that will not contain a lot of salt, because this can affect the way that your data is collected. But once you've got your sample and it's pure and it's clean, you can apply it to a grid. These are usually carbon grids with some sort of like metal foil, maybe gold, and you will apply it to the grid, hopefully evenly spread. And once this is applied, you want to flash freeze it. And when you flash freeze, you'll do this in liquid ethane. And this step is what they won the Nobel Prize sort of for incorporating into cryo-EM data collection. Because when you flash freeze in liquid ethane, you're not allowing the water in your solution to form crystalline ice, which could affect your data collection with electrons. Instead, you form a thin film of vitreous ice, which will not affect your data collection. And it kind of revolutionized this technique and made it a lot more accessible for people, and sort of started this cryo-EM revolution, which is what people were calling it. But yeah, once your grids are frozen, you can put them on the microscope and collect your data. It does actually look like this. So, the electrons will hit the grid, and you get this basically a projection, or like pictures, which we call micrographs, of the grid. And all of these gray dark spots are going to be what you're looking for. So maybe it's your virus, maybe it's your protein, and it will have some sort of difference between that and the background. And then once you've got this, you can pick your particles. And so looking at your grid, maybe you have a top view of a protein, which looks like a triangle. And so you can sort of pick this out in your solution. Maybe on its side, it would have one of these, I don't know, circular shapes. And so you can pick those particles, and then you'll have sort of two bins of your protein in different orientations. And once you have sort of got your 2D classes, which is what you call these bins of your view of the protein, you can then use this to make a 3D map. And then hopefully with some further refinement, you'll get a 3D model. And so that's sort of the overview of cryo-EM. It's much easier said than done. And like I said, I have done some cryo-EM as part of my PhD. And so I thought I would include a bit of real data from this. And so this is what those 2D classes look like. As you can see, the resolution is not great. But the protein that I work on looks a bit like this. This is a X-ray crystallography structure that I collected back in the first year of my PhD, actually. And you can see that the protein sort of has this heart shape when you're looking at it from this view. And so when I was looking at the grids, I was sort of looking for something that had a heart shape and maybe had a little bit more density down here, because this is where we have the two parts of the protein coming together. And you can actually see this in some of these classifications. So for example, this one, this is likely the protein looking head on. There's another one. This is probably it looking from the bottom up. And this is probably also a side view. And then we also have it from the other direction. So probably something like this would be looking at it from the side in this direction. And so from these 2D classes, we were able to sort of bin them and get a good view of the protein in all directions. Because you don't want to just have one view of your protein. And this is sort of another limitation that people might come into when doing cryo-EM, is that maybe your molecule will have an orientation in which it favors on the grid. And so you can only see it perhaps from this view. And this results in a lot of difficulty when trying to get a 3D structure of your protein, because you can only have a true understanding of it from one direction. But luckily here we have a really good spread of orientations of the protein. And so moving on from those 2D classes, we got this, which is called the ab initio reconstruction. And basically what this means is the software that we use was able to use these classes to make a 3D model of what it thought the protein looked like. And so it has this 3D model, and then these images here are 2D projections of the 3D model. And as you can see, it has this really nice heart shape, which was great to see because we know that this is actually the protein that we're looking at. And we see this sort of higher density here, which is great, like I said, because the protein that we look at is a dimer. So it means that each of these sections is separate and they overlap here. And so likely the density is going to be higher. And we see that in our top-down view, and we also see it in our two side-on views. And so when we look into the 3D structure of what the software thought that the protein looked like, this is what we got. This is a very early step of refinement. This is where it was back in November, and it's been running on the big computer since then a few times, and we're able to see some secondary structure. But it was just great to see that we had this nice heart shape, that we had, you know, a good understanding of what the protein could look like. And like you can see, this is how it looks. And then this is the structure that we got from the side, which once again looks like it could be correct. And so this was sort of a real example of cryo-EM. Obviously, this is a very low resolution structure. You really wouldn't be able to design anything based on this, just because it's just a big blob. You don't know, you know, where particular residues are. You don't know where its active site could be, where it's carrying out its functions. And so from this particular structure, you definitely wouldn't be able to make anything. But with further refinement, we're most likely going to get something which will give us a bit of insight into the structure like this. But like I said, X-ray crystallography is my favorite technique. This is what I'm doing my PhD with. And so basically, if you're looking at your electromagnetic spectrum, we're going to be working within this 10 to the minus 10 wavelengths, which is the X-ray part of the spectrum. And at this wavelength, we have a frequency of around 10 to the power of 18 hertz. And this allows us to see molecules at atomic resolution. And so this was kind of one of the first biological techniques used to get higher resolution understanding of structures. And like I said, my PhD is split between Oxford and Diamond Light Source. But I suppose not a lot of people know what diamond light source is. I didn't know what it was when I was not here. Um, and so this is diamond. It's the UK national synchrotron. It's in Harwell, which is about an hour from Oxford. And it's basically this huge ring. It's more than half a kilometer in its circumference, and it's basically a way of making X-rays. And I'll show you how we can do that using the ring structure. And so this is what the ring looks like kind of on the inside. And so the first part is that we have our electron gun, which is the beginning of the X-rays sort of life cycle. And in here we have a high voltage cathode. And at Diamond, this is tungsten. And it's heated under vacuum, and it gives electrons the sufficient thermal energy to sort of evaporate from the surface, and then we can have escape of electrons into what we call the LINAC, which is this part here. And so the LINAC is called that because it's a linear accelerator. And so we're going to take our electrons from our electron gun, we're going to push them into the LINAC, and here we're going to accelerate these charged ions to like a really, really high speed by subjecting them to a series of oscillating electron potentials along the linear beamline. And so at Diamond, they're going to be accelerated from 90 kilo electron volts to 100 million electron volts, which is 100 mega electron volts. And this is done using radio frequency cavities within the walls of our LINAC. And these are basically just big metallic chambers that contain electromagnetic fields. And these just force the electrons to be accelerated down this straight line. And then once we have our electrons at 100 mega electron volts or 0.1 giga electron volts, we put them into our booster ring. And from here, this actually looks like a perfect circle. The real booster ring isn't a circle, really. It's what I would call similar to an athletics track, where it sort of has two straight sections and then two curves on the side. And in the booster ring, this is just another way that we can get our electrons to be accelerated. And so at Diamond, there's 36 dipole bending magnets that are used to curve the electrons around the two bends on the side. And then on the two straight sections, we have, again, radio frequency voltage sources, which are used to accelerate the electrons on those two straight sides. And so we have these bending magnets, which allow us to accelerate in all directions and go around the corners. And by doing this, we're going to push our electron energy from 0.1 giga electron volts to 3 GeV. And this is a huge amount of energy, which then pushes these out into our storage ring. And so at Diamond, our storage ring has 25 straight sections. So yeah, this is kind of more realistic here because it's not really a perfect circle. It's more like short straight sections with little curves, where we have large electron magnets, which are called dipole magnets or bending magnets. And these are used to curve the electron beam in the space between these really short adjacent straight sections. And here we just maintain our beam at 3 GeV. And this entire ring is maintained under vacuum, just to stop electron scattering off of M molecules, because this would be pretty bad. But when the electrons are in the ring, and they're going around at 3 GeV, they complete the circuit in around 2 millionths of a second. And so that's nearly 7.5 times around the Earth's equator in 1 second. And so the electrons are moving extremely fast in the storage ring, which is this one. And so this is nearly the end of the life cycle of the beam. So what we have is this insertion device, and this is basically used to generate synchrotron radiation from our accelerated electrons. Within the insertion device, we have two main things. We can either have wigglers or we can have undulators. And both of these use alternating magnets to force the beam to trace a wave-like trajectory. And this is what generates our synchrotron radiation. So basically, our electrons are going to be moving from the storage ring into this insertion device where we have our magnets, which are going to be pushing them to form this wave. And this is our sort of synchrotron radiation. And then we're going to go into the beamline, which is where we're going to have our experimental setup. So, oh, sorry, this is the insertion device. And we can see that it's moving in this sort of like wiggling wave-like pattern. And that's what the magnets are doing for us. But once we have moved out of the insertion device, we're going to move into the beamline, like I said. And so the first part of the beamline is what we call the front end. And this is just used to channel the synchrotron radiation into our experimental area. And this is fitted at a tangent to our storage ring. And so it's kind of a way that we can get the electrons from moving in this circle to come off in a kind of easy way. And yeah, so the front end is basically just going to channel the synchrotron radiation. And then we have our optics hutch, which contains optical devices, sort of like mirrors and diffraction gratings, which are going to filter the beam so that we can have it at a particular wavelength that we would like for our experiments. But also we want to focus it so that we have complete control over where it's going, because you do want to direct it at your sample and hopefully not anything else. And then the final part is our experimental hutch. And so this is where we have our sample, like I said. And so here we're just going to put our sample on a little rotating arm, and it's surrounded by detectors. And so once the sample is hit with our X-ray, it's going to diffract off and we can collect it at particular angles. And so, like the name suggests, with X-ray crystallography, the one thing that you do need is crystals. And so a crystal is basically an ordered set of molecules. And so maybe you have the protein sitting end to end, and they form this very ordered, tight structure. And within this, they will crystallize and will have a nice ordered structure that we can get a diffraction pattern from, which can then be converted into our protein structure. But like I said at the beginning, getting crystals is quite difficult. Conditions are hugely ranging. Proteins are very temperamental. They sometimes like conditions one day, you change the pH 0.5 and they'll crash out of solution and they don't want to be there anymore. And so getting the crystals is sort of the first main barrier for X-ray crystallography. And so there are several different types of crystals that you might get. This is maybe like the starting point for crystallography. If you're just trying things out, putting your protein into what we call a screen, which will be a big plate of varying conditions in all directions, and sort of getting an idea of what things the protein likes. Maybe it likes sodium chloride, maybe it likes to have some sort of tris buffer, which allows it to be at a certain pH. And then from these, you can kind of get an idea of what the protein likes. But most of the time, you end up with something which we call a sea urchin. I mean, I would never take one of these and mount it on a beam, but it is a great starting point for getting your crystals. And so once you have something here, like I said, maybe the pH is like 6.5, and you think, okay, we have some sort of crystalline material here. What if we up our pH or we lower our pH or we increase the salt concentration or something like that? And maybe it will take you from a sea urchin to a needle. And honestly, needles, crystals, I feel like they get a lot of bad rap, but they're usually okay. I mean, they're kind of all clumped together here, but if you can separate them and, collect some data off of one of them, you might end up with a really good data set. And so one thing I would say with crystallography is that you shouldn't always judge what the crystal looks like, as being, and something that can tell you how good it's going to diffract and how good your data is going to be. I mean, I wouldn't take a sea urchin, I will say that, but a needle crystal, I'd be very happy to mount on a beam. But then maybe you want to get a little bit better. You want to have some separate crystals. You want to have them be quite big. So you might end up with something like a 2D plate. It's going to be very flat, maybe a little bit difficult to take off the plate. But then you may also end up with some microcrystals. There's no scale bars on these, but this could be something between 5 and 10 micrometers. So that's 10 to the power of minus 12 centimeters, which is 1 micrometer. And so very, very small. And so these might not be great for mounting a crystal, but they could be good for other different types of crystallography. But then really the final thing, if you really wanted to optimize your crystal setup, would be to get a thick 3D crystal, something which looks like this. I mean, these are great crystals. My crystals do not look like this. Maybe this is the dream. But once you have your crystals, you can put them on your beamline. And so, yep, maybe you have like a really nice 3D crystal like this. And what you're going to do is you're going to take them and you can put them on a loop like this. So when I say fishing for a crystal, you are truly fishing for a crystal. So you'll have a nylon loop like this. Maybe it's about 50 microns in diameter. And you're just going to go into your small little plate, looking down the microscope and fish it out, literally as if you're fishing. And then once you've done that, one thing you do need to do is cryo protect your crystals and cryo cool them before they go on the beam. And so one way to do this is to just put a viscous solution on them or something which is going to keep them surrounded before you plunge them in liquid nitrogen. So maybe something like glycerol or polyethylene glycol. And these are just kind of like thick, sugary solution sort of things, which will protect your crystal from getting radiation damage once you put it on the beam. But like I said, you don't always have to look at your crystal like this. You could look at it on something like this. This is a grid that we have at Diamond on one of our beam lines, which is called I-24. And this is our grid setup. And so maybe you have some microcrystals, and so you can just pipette them onto the grid, take away some of the liquid, and then mount them up onto the beam. And then your X-ray will scan across like this, hitting each crystal one time, and collecting your data like that. So these are kind of two ways you can collect X-ray crystallography data. Of course, there's several others, but I don't really have time to go into all of those. But once your crystals are mounted in the beamline, you want to take them, hit them with the beam, and you'll end up with something like this. And so this is our diffraction pattern. Once the X-ray beam is directed at the crystal, the X-ray will hit it, and then it will diffract off of the crystal. And it will have both constructive interference and destructive interference. But we will collect constructive interference as this diffraction pattern. And the position and the intensity of each of these dots is going to give us an idea of what our protein look like. Luckily, we have great ways of doing this now with computer techniques, because I could not even imagine how people did this back in the day by hand. But I guess the question is, how do you get from these to this nice structure? And so I'm not going to go into too much detail on this because it is quite an intensive process. But it's kind of split into two main stages. The first thing is integration, and this is the process of extracting the intensity information from the spots in the image. Sorry, I think the picture is supposed to come up. And so basically this step just involves four main things: spot finding, indexing, refining, and integration. So here the computer program will go through and it will either look at one image or it will look at several images from your data collection. And it will have a sort of intensity cutoff, and it will be able to pick out all of these black spots and also compare them with other spots in your other images to kind of separate them and know which ones are giving us information about our protein structure. Because we may also have something diffracting like water or some sort of condition that we have that could be contributing to these spots. But once you found your spots, you can do some indexing, which, like it sounds, you get a 3D index for each of your reflections. So you'll get three unique points for this, which are given three indices, H, K, and L, which you call your Miller indices. And it will give you a good approximation of your crystal's unit cell, which is sort of the way that it's arranged and how many sort of protein molecules you have within each small unit of your crystal, as well as the mosaicity, which is just another parameter with our crystals. And so once we have our indexes for each reflection and all of our spots picked, it can use a number of approximations and sort of get a true value for each of your positions. Because during indexing, the program is going to make a lot of approximations in its calculations, and the parameters it will report for your H, K, and L will be approximate numbers. They're not going to be exact. And so by measuring the intensity of the spots accurately on each image, we'll optimize that using the process of refinement. And we'll do some positional refinement, which will just minimize the difference between the observed and calculated positions of our spots on the detector, as well as post refinement, which kind of relies on the measurement of intensities of different partials belonging to the same reflection between different images. And using this information, we can basically get a precise center for each reflection on our images. And then the final step in this integration stage is integration. And once again, it's just about removing background noise from the spot intensity. So once you've done all of these steps of your integration pathway, you just end up with a true understanding of where each of your spots are on your diffraction pattern. And then scaling and merging is the next step of our processing. And it's basically just about putting the measured intensities onto a common scale and allowing for sort of imperfections in the experiment, and merging symmetry related measurements so that we can get a true understanding of the structure once we put it into a program. Like I said, it's a bit much, but this is kind of like a little bit of an idea of what we'll do with our diffraction image, and then we can take it into our protein structure. All the programs do this for you right now, so there's not much that you actually have to do. You can give it a model that it can base it on, which makes it a lot easier because it can project a diffraction image of what it thinks it could look like and then kind of look at the difference between the two of them. Or with the production of AlphaFold, which I'm not really sure if you know what that is, but it's a program made by Google DeepMind, and it can predict protein structures from their linear code. And using predicted models, you can use this as a way to get your real structure by using it as a sort of model to start off of. But yeah, this is kind of the process of crystallography. We'll do our phasing from our diffraction image, end up with something which we call an electron density map, which is this blue stuff. And so it kind of gives us an idea of what sort of information we're getting from the pattern. And then we can fit our structure into this diffraction pattern like this. And then we'll end up with a nice final protein structure. We go through several rounds of refinement back to this diffraction pattern and go back around again. But in the end, you end up with a nice protein structure, and you can use this for moving into your drug discovery. And so, yeah, the first thing that you want to do once you have your protein structure is think about the function of your protein, and some important areas which might be involved in this. So yeah, like I said, this is the protein that I work on. And here is its active site. And so this is the area that we want to target when we look at our protein. but I thought we could just do a little bit of an overview of what we'll do once we have the structure. Because there are a few things that you can do, right? This is kind of the basic idea of, drug discovery from protein structures. We'll think about our target. So like with the protein that I look at, this is involved in COVID. And so it's a nice choice. Then we'll get our structure and look for binding sites. And then you can either do some in silico techniques, so some computer docking, or you can do it experimentally. So yeah, molecular docking, that's a way of doing it within a computer, or molecular dynamics, another way of doing it in the computer. And then experimental techniques such as doing it by hand, getting your crystals and soaking them with drugs, and then seeing, what binds and what doesn't bind. And once we sort of have an idea of, our protein structure where we have binders, we can then move into the next step, which will be in optimization of the lead compound that we find and basically making it a better binder and a better drug. And then you have your drug, and hopefully you can take it into clinical trials. And so that was all kind of an overview. We've got an idea of how we can get our protein structure, and then the steps that we want to take once we have our structure and moving it into a new drug. And so I thought I would give you a couple of examples of when this has actually been used, because in theory, I suppose it sounds quite easy, but in the end, it actually does take a very long time and a lot of work. So here is an example. Sorry, it's not moving. Here we go. And so this is of the drug called Saquinavir. And this is used to target HIV and AIDS. And the protein target is an HIV protease, which acts in a similar way to the protein that I was talking about earlier, as it cleaves other proteins. And so it works in the step right here, where it cuts up your polyproteins that are produced from your viral DNA, and it cuts them all up into smaller functional units, which can then go on to produce new viral particles. And so if we want to target that protein, we're not going to be making more viruses, which is ultimately good for disease progression. And so what they did was they got this structure of the HIV protease, and it looks a little bit like this. It's similar to the protein I was talking about before, in the fact that it's a dimer. And so it has these two protein units, the pink one and the blue one, which are identical. They each contain 99 amino acids, and they have this active site in the middle of the two of them. And this protease is a member of the aspartic acid protease family, which means that its catalytic site contains asparagine, threonine, and glycine as a trio of catalytic residues. And we have this central cavity where our other proteins will be binding, and we'll have sort of cutting up of the protein. And so they use this structural insight to give us an idea of how to move forwards in targeting the protein. So it was based on this observation that it cleaves sequences containing dipeptides that look a bit like tyrosine and proline, or phenylalanine and proline, which are basically amino acids which have sort of benzene-looking side chains, especially tyrosine and phenylalanine. And they made this drug Saquinavir, which is an aspartic acid derivative, which, if you can remember from back here, the active site here contains that. And so this is our aspartic acid. And then they added this DIQ group, which was used to improve its solubility as well as its binding potency to the protein. And from sort of these subunits, they made this drug, which looks a little bit like a peptide. If you look at the backbone, it has these amide and carbonyl groups, which is similar to a protein backbone. And then they have this phenyl group, similar to the phenylalanine. And then, like I said, they add the DIQ group into here. And so from this, they had like a nice X-ray crystal structure of the protein bound to the drug. Um, and so they were able to get some really nice structural insights into the way that it interacts with the active site. Did I have a, sorry, I thought there was another structure on here of that. But you can kind of see it here, where you have this hydrogen bonding between the amide carbonyls in the backbone and this water within one of the parts of the protein structure, as well as some interactions with other residues which form this cavity. And so this was actually the first protease inhibitor ever approved. It was approved back in 1995, and it sort of formed the the cornerstone of the antiretroviral therapies. One thing to note is that it is boosted with ritonavir, and this is just a way to get it to not be broken down. It kind of increases its lifetime in the body, because that's the thing with a lot of these small molecules is that they will be broken down quite quickly. And this is a CYP3A4 inhibitor. And so that's one example. Another example of targeting a protease, which is close to my heart, is targeting the SARS-CoV-2 main protease, which, like I said, is the protein that I work on. And this also plays a vital role in processing viral polyproteins. And so this is the life cycle of our SARS-CoV-2 virus when it enters the cell. And so it sort of enters the cell here. It releases its viral genome, which is single-stranded RNA genome. And then it's translated into these two polyproteins called PP1A and PP1AB. And this translation is actually done by host cell material. And then it creates these two polyproteins. And then we have two proteases, one which is PLpro, the papain-like protease of SARS-CoV-2, and then Mpro, which is the main protease. And these cut this up into several non-structural proteins, which can then go on to produce this replicase transcriptase complex, or also go into making structural proteins, and ultimately leads to the assembly of new viruses and their release. And so this is a really key part of the viral life cycle, as it acts really, really early. And so inhibiting its activity will sort of cut off the rest of this and the production of new viruses and sort of the infection. And luckily, it has no human homologs as well. And so it really doesn't have many off-target effects on human proteins, which is a really big win when looking into making drugs based on proteins, because a lot of the time there is a bit of overlap. And so when creating this drug, it was all done in a big, quick sort of rush at the beginning of COVID, where people really wanted to make drugs and sort of stop the pandemic as soon as possible. But very early on, the crystal structure of Mpro was determined using X-ray crystallography. And so this is what the protein looks like. Like I said, it's this nice heart shape. It's made up of two monomers, which are these two parts here, each of which contains these three domains. So we have domains 1 and 2, which are involved in forming our active site, and then domain 3, which makes it a dimer, because you have these two bundles of alpha helices kind of coming together, and this actually allows the protein to work, because when it's in its monomeric form, it has no activity at all. And so they kind of determined that here between the two domains, we have our active site, which is composed of a catalytic cysteine, and our general base, which is going to be our histidine. And so these two act to cleave proteins. And then also we have these sub-pockets which were determined in the active site. And so these are key parts of the site which allow for substrate recognition. And by that, I mean things that will bind in there. And so using these structural insights, they were able to rationally design a drug to bind in here. And so they made this drug. It's called nirmatrelvir. And this was made to covalently inhibit Mpro, and it does this by forming a reversible covalent bond with our catalytic cysteine. And so the drug contains this nitrile warhead. It's just like its key point. And it basically forms this really nice covalent bond with our cysteine molecule here at this sulfur group with this nitrile. and we get this really nice binding here, and it stops it from having activity on cleaving other proteins. And so they made some modifications to this as well to optimize its binding affinity within the other subsites, and to kind of, so like I said, nirmatrelvir was rationally designed to covalently inhibit Mpro by forming this reversible covalent bond with the cysteine at this SH group, and then with our nitrile within nirmatrelvir. And so this makes this really nice covalent bond and basically stops any other proteins from getting into the active site where it could be cleaved. And so nirmatrelvir was FDA approved back in 2022. So it was a little bit after the pandemic finished. I think small molecules take much longer than obviously vaccines take to develop. But this is a really effective drug. And you can see that it sits in the active site really nicely here. It forms a covalent bond here with the cysteine, which is somewhere under here, but it hasn't, this is a non-bound version of it. And then it has these projections into each of these subsites. This S1 prime subsite is a really key part of the binding site because we have what we call an oxyanion hole, which helps to stabilize it within its steps of cleaving. And so targeting this is a really great place, as well as these two other pockets where it sits really, really nicely. And so nirmatrelvir, like I said, was made back in 2022. And then recently they made this second generation version, which is called ibucetrelvir, where they made these two modifications. They both still contain this nice electrophilic nitrile group, but then they added these two regions to enhance its binding affinity to Mpro, but also to improve its metabolic stability, and to allow it to be taken without the administration of ritonavir, which nirmatrelvir does require, similarly to saquinavir. And so it also improved its safety profile, and this, I'm pretty sure, was FDA approved last year. And if it hasn't been FDA approved, it will be approved very soon. But those are sort of two examples of structure-based drug discovery. One cool thing about ibucetrelvir and nirmatrelvir is that they kind of have this pan protein binding and inhibition activity. And so this is its binding into SARS-CoV-2 Mpro, but then it can also bind into the MERS-CoV Mpro active site as well. And so we can kind of think about using some of these drugs as pan coronaviral targeting drugs. I don't think they've been approved for MERS or any of the other coronaviruses yet, but it kind of is a nice way to start as a scaffold for making drugs which target these, perhaps just like modifying them a little bit to affect the particular residues which are in each of the subpockets in the different proteases. So yeah, those are two examples of structure-based drug discovery, mostly based on experimental methods. But it felt kind of stupid to talk about drug discovery without talking about the use of AI and machine learning and in silico approaches, which are really common now. And so there are a few computer-aided drug design methods. So like I said, you have AlphaFold, which can predict structures. And then there's also molecular dynamics simulations that you can do. You can get some ligand docking going where you just see if you can have different ligands binding to your protein, sort of in an algorithm where you have parameterization of your protein to kind of make it act like it would act in real life within your computer. Again, you can also do this with a lot of other things, and machine learning and sort of using a test set really helps in kind of getting an idea of what you can start with before moving into experimental techniques. Because although these AI methods are really useful, you still need to have real screening, hit identification, and experimental techniques to complement your sort of predictions.